So thank you very much for the opportunity to present our work. And thank you to Katherine Brown and Alexi for putting together this seminar series. As Katherine said, I'm a research fellow at the University of Padua in North Italy. And today I'm going to talk about biomechanics of neural tube morphogenesis. So I'm interested in neural tube closure, which is the embryonic precursor of the brain and spinal cord. And the reason why we are working on this is because it's a beautiful morphogenetic process, but it also has clinical relevance. And to illustrate this point, here you can see, hopefully you can see my cursor as well. You can see neural tube closure in the chicken embryo. This is from a transgenic line that we used this summer at the ECT workshop up in Edinburgh. And the neural folds in this process approach the midline and fuse. And this process continues like a zipper from the midbrain region that is just outside the field of view here, all the way to the bottom of the spine. So along this way, different neuropores or gaps are formed along the embryonic axis. For example, here at the back, you can see the posterior neuropore with this elliptical shape that needs to completely close. The whole event takes place within 14 hours in the chick, and if it fails, it leads to neural tube defects, hence the clinical relevance. Now, we are looking at neural tube closure not only from a genetic and molecular perspective, but also from a mechanical one. And this is because, as you can hopefully appreciate, it involves dramatic changes in shape. And to achieve these dramatic changes in tissue shape, you need mechanical force production and mechanical accommodation. So mechanical forces may be intrinsic to the neuroepithelium, which is this tissue here, and you can see it at different phases of closure, but may also be extrinsic. So they may originate from other tissues, such as the surface ectoderm, which is a superficial layer on the side, and this will be the future skin, presomatic mesoderm, and other tissues. But on the whole, pro-closure forces, which are here in green, need to counteract anti-closure forces for closure to be successfully completed. Now, we and others have worked on cellular force generating mechanisms, and these have been described across scales and in different organisms. For example, in the mouse embryo, here you can see we are looking down at the spinal region of an embryonic day 9 mouse embryo, and we have described apical constriction as a mechanism the cells employ to generate force. This apical constriction is in the neuroepithelium. And as you can see here, when we go in and we inhibit the machinery that controls apical constriction by ROCK inhibition and culture, we first see that the posterior neuropore does not close properly. It's very spread and abnormal. And this is also associated with larger apical areas of the neuroepithelial cells. So we have this link between failure of closure and these larger apical areas in the neuroepithelium. Then if we go to the other side of the mouse embryo, to the forming brain, there's another neuropore called hindbrain neuropore, where the force-generating mechanisms are employed by a different tissue, the surface ectoderm or future skin. So in this case, you can see here the surface ectoderm stained by ECAT here. We described actomyosin cables that start very long, about 600 microns. They are very rich in protrusions, as you can see here with phalloidin staining. And then they constrict to bring the neural folds together. So actin cables are employed by the surface ectoderm. And when we go in and cut them, we see that they're under high mechanical tension. So if you're interested more in this, you can check our paper, but we are about to also publish a preprint in an ongoing story of this version where we're trying to model very precisely the cell shape changes that the surface ectoderm goes through during closure in this case. And it's in collaboration with Fernanda Perez Verdugo, Sila Banerjee, and Gabe Gallia. Now, as I said, these are some mechanisms that cells can use to generate forces. Others, for example, are convergent extension or cell division and are described in different organisms. But one very difficult thing to actually go and pursue is not only how the cells generate the forces, but how much these forces are, what is the magnitude of these forces. And in order to answer this, we came up with the idea of designing force sensors inside the neural tube of the embryos. For this, we switched to the chicken embryo. And this is because these embryos are flat in early development and can grow well outside the body of the mother. So we thought we would like to find a way to construct force sensors inside the neural tube with attachment to the neural folds. And then the expectation here would be that as the neural folds approach each other and closure progresses, these sensors that are spring-like would deform. And based on this information, we'll be able to calculate the mechanical forces that the neural folds produce. And in order to achieve this, we are using a polymer that was designed and first described by my current lab at the University of Padua. And it was first described for 3D printing structures, millimeter-long structures under the mouse dermis. It's all based on photocross-linking. So basically, this polymer, as you can see here, is a conjugation between coumarin derivatives and PEG hydrogels. And then by using a 2-photon laser, we can 3D print the structures we want in situ at the specific anatomical location we want and with micron level resolution. We can get as low as 1 micron, as accurate as 1 micron resolution. So this technology, as I said, was described in 2020 for very long structures, but then we wanted to see how this chemistry will work in our case inside the chicken embryo. So we went through many different iterations of characterization because this is a really different environment inside the embryo to make sure that the chemistry is working. And the experimental setup we ended up with is actually not difficult, it's quite straightforward. So the polymer, before it's cross-linked, is viscous. We add it as a drop inside the neuropore of the chicken embryo. And then with a 2-photon laser, we can 3D print whatever structure we want. And after that, we can go in and do atomic force microscopy inside in vivo and try and understand what kind of stiffness we can achieve. This showed us, in collaboration with Monica Zomo and Anna Ortsolo, that we actually have 2 different levels of control of stiffness. The first one is laser power. The higher the laser power, the stiffer the polymer we get. And the second one, because the rate of cross-linking is increased. And the second one is concentration. And this is because the higher the concentration, the higher the availability of the photoactive groups. So that gave us a very powerful tool to be able to start and printing force sensors. And once we managed to confirm that the chemistry is working and we have this fine control of the polymer in vivo, we went on to many iterations in order to approximate the spring. So first we started from simple shapes, as you can see here, like simple bars, which have the advantage of they're very easy to print, and you can also print many different bars along the axis. So the idea also here was to try and characterize the different forces along the anterior-posterior axis. The problem, however, with the bars is that a lot of force is being concentrated at a very small region, and then we saw that even within half an hour, the bars were pushed out of the embryo as closure progressed. So that was not very useful for us. And then we were creative. We moved on to different shapes. For example, here you have this accordion type shape with 2 stiff bars parallel to the neural folds and 2 V shapes in between. And as you can hopefully appreciate, in this case, we did have deformation, but the force deformation relationship was highly nonlinear. So it was not easy to use this shape further down the line to model the forces and calculate the forces. And after that, we switched to a simple cylinder that has the advantage of being very able to reproduce. So imagine this ring, but it also has a Z thickness of about 15-20 microns. And it is easy to adapt, so it's adaptable between different embryos, because obviously there's biological variability, so you don't have exactly the same geometry of the neural tube between different embryos. And in this case, in collaboration with Sylvia Todros and Piero Pavan, we performed finite element modeling based on biological data. So here you can see these 2 rectangles that represent the neural folds, and in the middle we have a cylinder that is compressed in X and expanded in Y. And finite element modeling, FEM, produced a parameterized equation where there is a linear relationship between force and deformation of the cylinder. And here the E is the elastic modulus that we can measure, as I said, by AFM. Now, what we also saw from this modeling iterations was that there's an inverse correlation between force and deformation. So the smaller the diameter of the cylinder, the more force is required for the embryo to deform it. So moving on, after we had the characterization of the polymer, the chemistry, and the modeling in place, we started detecting some meaningful deformation. Here you can see a chicken embryo 20 hours post-printing. The magenta lines here indicate the neural folds that have zipped anterior and posteriorly to the cylinder, but have remained open directly above it. And then on the right here, you can see maximum projection from confocal images of T0 and T20 hours. We start with a circular shape, and then within 20 hours, that this shape, because of the compression of the neural folds as they close, has become elliptical. And on the right, you can see the approximation of forces in these very late time points. And this is a range between 50 and 120 nanonewton. So yes, these are very late time points, and it's only an approximation of these states, but this told us that we have a polymer that is deformable, it's adaptable, and also it stays inside the embryo and it's fully biocompatible. So then we combined this technology with live imaging, and here you can see an embryo closing down in about 2 hours. The embryo is stained with cell mask, and if we move on here and see the overlay, again, we have this compression of the cylinder over the 2 hour period. And this makes sense because, as I said, the cylinder is attached and it's compressed mediolaterally. And then when we try to convert this deformation into force, we see that it's increasing linearly over time, and it plateaus reproducibly at about 90 minutes. And this is the same behavior in different embryos. So about at least an N of 10 we have here that showed the same reproducible increase and then plateau. And for the last challenge, we tried to modulate the mechanical forces. So as I said, apical constriction in the neuroepithelium creates force to bring the folds together. So when we go in and use pharmacological ROCK inhibition, we have this kind of image here. The neural folds are springing apart over time, and together with the neural folds, the attached cylinder is stretched mediolaterally. And then we can see the overlay over here with the magenta being the 2 hour shape. And then the corresponding force, which is this case is negative because the deformation is opposite to the direction of closure. This tells us on the whole that the active anti-closure forces are in place, because if we didn't have active anti-closure forces, we would just see the neural folds staying in place, stop closing, but staying in place rather than reopening. And bringing all this together, we showed that we are able to perform in situ fabrication of force sensors, and this enables dynamic quantification of mechanical forces in living embryos in absolute numbers. Then the closing neural folds generate over 100 nanonewton compression force. And then if we diminish proclosure force by pharmacological ROCK inhibition, we can reveal active anti-closure forces. And just for my couple slides in the end, I just wanted to touch upon what we're trying to do now. So as I said, one of the advantages of the technology is that we can really control the thickness, the Z thickness of the shapes that we're printing. So here you can see, for example, the cylinder that I was just describing, which has dorsal attachment, the open neural tube, but also we can print very thick ones that we can attach all the way to the floor plate. And then, I mean, this orientation will be a bit different than before. You can see the anteriors of the head on the left, and on the right, you see the spinal cord. When we try to do that, we saw this deformation of the cylinder that we think comes from the floor plate itself. And then if you can see here at 120 minutes, we have this bulging of the tissue along with our shape. And we think that what this reveals is tissue flows in the open neuroepithelium. And now we're trying to, first of all, visualize properly and quantify these tissue flows, for example, by PAV, and then trying to understand what their role exactly is in closure. And one more example of this, here you can see 2 half cylinders that are printing along different levels of the axis. And we can actually visualize 2 different behaviors. In the more anterior parts, the next day we see compression of the cylinder, whereas in the more posterior part, we see rotation of the cylinder in the XY dimension. And this also potentially shows us differences in the flow in the right and left part of the embryo. So this is something we are following up now. Hopefully we can discuss about it in another seminar. And brings me to my last slide. I would like to thank my current lab, the lab of Professor Alvasore at the University of Padua, Gabe Gallia at University College London, which is where this project started, and our collaborators from Carnegie Mellon and Leuven. And thank you for your attention.
Great. Thank you so much, Eirini. That was fantastic. So if anybody has any questions, please do put those into the Q&A box so that Eirini can answer them. While we're waiting for questions, so we've got one question in on the chat already, which is about the cellular source that's generating the forces. Is that presuming that's within that closing epithelium, are there specific leader cells that are generating the force? Is it coming from further back within the tissue? Do you have a feel for how that's working? And can these kinds of technologies help you to answer those questions?
Yeah, so, well, I do have an answer coming from our work on the mouse hindbrain neuropore, where we saw that we have this directed crawling. So the surface ectoderm cells synthesize their own matrix and they crawl towards the center of the neuropore directionally. So this creates a force that is required for closure. Therefore, we have these leader cells in the first row, which are the same ones that create the cable, and they're the ones that create the force to close. We can use this technology to answer different questions. With respect to the surface ectoderm, apart from making barriers and seeing how these are deformed along the superficial layer, I cannot really think of anything else, but the technology in general is very versatile.
Great, thank you. We have one question in the Q&A from Heather Etchevers, who says, great presentation. Might these sensors induce problems themselves in that they might interfere with the primary cilia towards the luminal part of the neural tube?
Yeah, so I haven't specifically looked at cilia, but I mean, I have a slide, but anyway, you can also see it in the paper. We have done a lot of characterization of actomyosin, and we saw that printing doesn't really affect actomyosin. We don't really see any abnormal accumulations or any differences in the apical region of actomyosin. And we have done quite a lot of characterization with respect to zippering rate, somatogenesis, axial elongation, and none of them is affected by it. Now, specifically about cilia, I haven't really looked, but in general, it doesn't look like we have any very obvious effects in closure or survival of the embryo.
And then I had a question as well, which was, and you sort of started talking about this at the end by looking at the kind of different positions along the anterior-posterior axis, but you said there's this sort of consistent plateau at 90 minutes. Is that 90 minutes regardless of where along the AP axis or when during the process you're putting it in? So is that something about the time at which it takes to reach equilibrium is not the right word, but it can't be deformed anymore or whatever? Or is that about the, I guess, is that about the embryo or is that about the sensor, that time to plateau?
Yeah, it's a very good question. So basically, all our experiments were at the part of the embryo where the neurofolds are parallel because we wanted to model parallel points. And along this area, it's always the same. It's always plateau at 90 minutes, which probably means that, yeah, it does produce the force for this time, and probably this is the time it needs to reach the other side. And then somehow it knows that it should stop. That's what we think. But it's something we force decay. It's something we want to follow up on an independent project.
Okay, thank you. And then one final question in the Q&A, and then we'll have to move on, which comes from Shailendra Singh, which is, thanks for the great work. How did you decide the threshold for polymer density that can affect the tensile strength of the polymer ring when you were making that?
So how do we,
how did we decide the threshold for polymer density that would then affect the tensile strength of the ring?
Well, out of a lot of trial and error. It's the short answer. Yes.
Great. Thank you very much, Eirini,